We had a discussion last time about the metabolism and energy production. So you can hear me, guys.
Yes, we can hear you.
Yes, clear.
So the aims of this lecture will be, we will discuss bioenergetics and we will discuss the oxidative phosphorylation. So these will be our objectives. We'll define it. We will explain enzyme system. We will express our understanding of the classification of the enzyme they are involved, like oxidases, oxygenases, dehydrogenases. We will describe the respiratory chain and oxidative phosphorylation, the process. We will describe the different types of the cytochromes. And then we will see what is redox potential and how ATP is generated. So these will be our objectives. We will have the 3 sessions of this, maybe 3-4 sessions. And in a nutshell, we will see all related things regarding the oxidative phosphorylation. Clear? If I summarize it here, what we need in our body, the body need energy to perform some jobs, to function. Clear? Energy is in the form of ATP. Clear, guys?
Clear.
Yes.
Yes.
Energy is coming from food stuff. Foodstuffs are 3 major foodstuff: carbohydrate, lipids, and protein. So if I make it, if we summarize it, we take food, carbohydrate, lipid, and protein. We burn food. It gives us energy. Energy, we utilize it for functions. Clear? Clear, guys?
Yes, clear.
Yeah, so far so good.
This burning is oxidation. Formation of this ATP is phosphorylation. Okay. So
Okay.
burning of the food is oxidation. In results of this burning, the ATP which is formed, this function is called phosphorylation. Together, we call it bioenergetics. Bioenergetics. Bio mana these components, energetics mana this component. Clear?
Yes.
Okay. So, what is bioenergetics? Can somebody read this?
Okay,
Yes,
I
we read.
can.
Yeah, go ahead.
Bioenergetics is the study of the energy chains combining biochemical reactions. Biologic systems are essentially isothermic and use chemical energy to power living processes. Metabolism is the process by which an animal can obtain suitable fuel from food.
Okay. So, number 1, it is the, thank you very much. Number 1, it is the study of the energy changes in the biochemical reaction. Number 2, it is the Basically, the system is isothermic. Iso means same, thermic means temperature. And then it uses chemical energy to power living process. All this what we discuss. These are chemicals, chemical energy, power. Clear? Isothermic. Here temperature was, say for example, 97 degree Fahrenheit. When you burn it, temperature will increase or decrease?
It will increase.
Okay. So, and then metabolism is a process by which animal can obtain suitable fuel from food. Clear? So all these three things that we covered in the first slide. So the term metabolism is described as highly organized chemical reactions. Organized one are 1, 2, 3, 4, 5, 6, 7, like this. Not random, huh? And the major role of the metabolism is to capture
Energy.
chemical
energy.
from food stuff. Convert into as ATP. And then this ATP is utilized for functions. Synthetic function, transport function, muscle work, whatever you function. Lungs function, brain function, heart function, kidney function, muscle function, kullu functions. Clear?
Yes.
Clear.
Okay, so metabolism is a combination of two things. We discussed this last time, anabolism and catabolism. We say larger molecules are broken down into smaller molecule, gives energy. This is catabolism, and then smaller molecule combined with the help of energy. This is anabolism. We said breakdown of protein to amino acid is catabolism, and synthesis of protein from amino acid and glucose to starch is the glucose to glycogen in the human body. Glycogen in the human body, starch in plants and glycogen in the human body. It is the synthesis of larger molecule. This is called anabolism. Okay.
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 And we discussed the difference between the anabolism and catabolism, and we saw that anabolism use energy, catabolism release energy. Anabolism is endergonic, catabolism exergonic. We said that anabolism, you use energy, you go like you are going uphill. Catabolism is as like you are going downhill very fast. Anabolism, simple molecule to form compounds, and catabolism, larger molecule to convert into smaller compounds. So
What does it mean by moving towards uphill?
you are going towards mountain. On the mountain, it is difficult. Coming from the mountain is easy. On the mountain, you use energy. Coming from the mountain, you don't use that much energy. Clear? Uphill, downhill.
Clear. Thank you.
Okay. Yes. Good. So, and this is what we have discussed last time: the exergonic reaction and then the endergonic reactions. This is what we have discussed last time. And then this was the one of the example where we discussed that glutamic acid is an amino acid. When it will combine with ammonia, it will make glutamine. And this reaction, when glutamic acid combined with ammonia to make glutamine, this is an
Anabolism.
endergonic reaction. Delta G is positive, which is 3.4.
Meaning
Then
it needs energy.
yes, then there is an ATP. ATP will help of water molecule. You know, we discussed this. If you remember the classification of enzyme, we discussed oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. And one of the example of the breakdown of compound was with the help of hydrolases of water. You break things with the help of water. You throw water on the compound and compound is break. Clear? You remember we discussed this last time in previous.
Yes.
So same situation is here. ATP with the help of water molecule. ATP is broken down. ATP is like this. This is the structure of ATP. This is ribose, this is adenine, this is ribose, and then this is one phosphate, this is another phosphate, this is another phosphate. So how many phosphate? Adenine, adenosine, triphosphate. This is ribose, this is adenine, which is a nitrogenous base, ribose is a 5-carbon sugar, and then these are 3 phosphates. So this makes ATP, this makes ATP. Adenosine triphosphate, adenosine tri. When you put water on it, you throw water on it, so this phosphate is removed. So when this phosphate is removed, how many phosphate will left behind?
Two.
So this is ADP. So ATP will convert into ADP. And this phosphate will be released as this.
As energy, yeah.
And we know that when this phosphate is released, it will give how much energy? 7.3
-7.3.
If you wanted to add it, for example, you want to reverse it. It is possible, sir. If you want to reverse it,
Yes.
you want to add phosphate with ADP, how much energy you need?
More than 7.3.
More than 7.3. So this is in the negative, that is will be in the positive. Clear? Is this clear?
Yes, clear.
Yes.
But what is this combined reaction? What is this combined reaction? If you combine these two, so delta G is -3. So what is the combined reaction? Is exergonic or endergonic?
Exergonic.
Exergonic.
Exergonic reaction. Clear? So this is how we can see this. So human being can generate ATP. We saw it, carbohydrate, fatty acid, amino acid. At the simplest level, energy homeostasis involves a balance between fuel intake and energy expenditure. If there is a balance of intake is to expense, then everything will be normal. If intake is more and expense is less, what will happen?
Obesity.
Obesity. Excellent. And if its intake is less,
Starvation.
Starvation.
starvation or muscle body wasting, weight loss. Come on. Good. Excellent. So we know energy. What is energy? We discussed this last time, that it is the amount of the heat which can raise the temperature of 1 kilogram of water by 1 degree Celsius. This is called 1 kilocalorie. So here we saw this last time as well: food, carbohydrate, and this is the ADP, and these are the hydrogen acceptors. So our issue is that how this is generated. We know that it's coming from food stuff. Up till now, we know that it's coming from the catabolic process. We know it is used in the anabolic process. But this is the main mushkila. You understand what I'm saying? We know that there is a marriage, there will be kids. But what is this? What is this process? How we get this ATP, adenosine triphosphates? Clear? So this is the main topic, that how we burn food and then how we get this ATP. Because this is the most important pillar of the human body. This is the most important thing which can run the body. So you are thinking ATP. You are walking ATP. Your lungs functioning ATP. Your heart functioning ATP. Mana, everything which is working in the body is under by the use of ATP. So what is ATP actually? So ATP is, as I said, adenosine triphosphate. Adenosine is a nitrogenous base. You know what are nitrogenous bases?
Yes.
Adenine, guanine, cytosine.
Good, excellent. There are two types, purine and pyrimidine. Purine are adenine and guanine. Pyrimidine are cytosine,
And
thymine, and uracil. Cytosine, thymine, and uracil. So this adenine, and they are present, this is so what we call it in combination. This structure is what? If you want to give a name, a scientific name, Huh?
Nucleotide.
Nucleotide. Excellent. Nucleotide. And nucleotide is a building block of
DNA.
DNA.
nucleic acid. Excellent. Nucleic acid. And nucleic acid are of two types. One is DNA, another is RNA.
Oh no.
We know up till now, and this is very clear. Clear? But
Yes.
in addition to this, that this nucleotide, which is a building block of nucleic acid, it is also an energy currency, like real. It is also an energy currency. Clear? Because the phosphate which is present in this nucleotide, they have too much energy. They have too much energy. Clear?
Yes,
So
clear.
we say that this is a cell energy or energy currency of the body. So this is adenine, this is ribose, and this is ester bond, esterification with one phosphate, with another phosphate, and with another phosphate. And phosphate which is present here, this phosphate, here's a bond. This phosphate is in the form of H3PO4. So this is H3PO4, basically phosphoric acid. Clear? And how you can write this H3PO4? Like this one, OH, OH, and OH. And then there is another one. OH, OH, and OH. Then this OH, they will make a bond. Water molecule will go out. And then there will be only one oxygen. And this oxygen is here. Is this clear?
Yes, clear.
So this is how the phosphate. So whenever you want to remove this phosphate, you need water, because you will throw water on it and this phosphate will be released. So whenever this phosphate release, this phosphate will perform, will give energy, and then that energy will perform function. Okay, so it's a cell energy and provides help to function of the body. So this is how ATP, this phosphate is released with the help of water. And I just explained to you how it will be, why it needs water. And then this phosphate is released. Inorganic phosphate is released. When it is released in the form of inorganic phosphate, then it releases 7.3 kilocalorie energy, and then it performs function. Okay, now the question here is that how we can obtain ATP. So we discussed last time that there are two mechanisms to obtain ATP. You remember? One, we said substrate level phosphorylation, SL. Another, we said oxidative phosphorylation, oxidative phosphorylation. So one is substrate level phosphorylation, another is oxidative phosphorylation. Clear? And I gave you the example of these two is the cash money and check money. You remember this?
Yes, we remember.
I told you that we explained this, that if you have a cash of 500, even 50 riyal, so you can at least buy a coffee. But if you have a check of 500 riyal, which is 10 times more than the 50 riyal, but this is in the form of check as a paper, so you cannot even buy a bottle of water. Although the amount is more. Although the amount is more. However, if you take this check to the bank, and in the bank you go to cash counter, and then in the cash counter that person will give you money, then this is too much money. This is 10 times more than this one. Clear?
This example is for the mitochondria, right?
Yes.
This example is for mitochondria. So this is what happened. This cash is the example of substrate level phosphorylation, where you get quick energy, energy at the site of reaction, energy at the site of reaction, but definitely this will be in a small, this will be in a small amount. Clear?
Clear.
On the other hand, this is example of, check example, is the example of oxidative phosphorylation. Here you will get more energy, but you have to take hydrogen from cytoplasm with the help of hydrogen acceptor to the mitochondria, and then in the mitochondria, which is a bank, you have to go to the cash counter, which is respiratory chain, and then in the respiratory chain, with a long process, it will give you the good outcome or good amount of the energy. So this is a long process. Is this clear?
Yes.
This is called oxidative phosphorylation. Oxidative phosphorylation, where there will be oxidation side by side, and we know that by oxidation we mean burning. So you know that mitochondria is the generator, energy generator, energy powerhouse of the body. So it burns the fuel, it oxidizes the fuel, releases the energy, and then energy is packed in the form of ATP, and then that ATP is given to the body, and then body use it for the function of lungs, heart, kidney, whatever you want. Is this clear?
Yeah.
Okay, good. This is called oxidative phosphorylation. The process by which it is carried out is called chemoosmosis. Chemoosmosis. And you know, if you remember the process of osmosis, it is the movement of the water from low solute to high concentration of the solute. Process of the water. And here, this is the chemical transport, chemiosmosis. So this is the movement of chemicals, particularly the hydrogen, Clear?
Yes, clear.
Okay. So now we'll go for some of the example of substrate level phosphorylation. So could anybody read this?
Yes.
Okay, so read.
Yeah.
Okay, a high energy bond is formed in the substrate while being oxidized. ATP is then generated at the expense of this high energy bond as the following reaction.
Okay. So can you explain these three lines? What you understood from these three lines?
Okay, what I know is this bisphosphoglycerate is adding to ADP, and with the help of the phosphoglycerate kinase, it adds.
No. Here, these three lines. That line that you explained.
Okay.
Ah.
So there
In
is.
your words, if I ask you that explain this in your wordings, how you will explain it?
Okay, that there is an oxidation reaction with a high energy bond that is substrate level phosphorylation. That's what I
Good.
understand.
Excellent. Anybody else? Mumtaz. Anyone can explain these three lines?
Yes, I think due to the oxidation, it results in a high energy bond, therefore giving ATP at the expense of the
If
high
I
energy bond.
ask you to explain it in Arabic, would you be able to explain it in Arabic?
I don't think I understood good enough to explain it in Arabic.
Sorry?
I don't think I understood it enough to explain it in Arabic.
Yes. Anyone else?
Yes, doctor.
Yes.
While oxidation, the energy bond will form.
Okay.
That will help the ATP form.
Okay. 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 So let me give you an example. Okay. What is the meaning of this thing? The meaning of this reaction is A plus B will make C. Clear?
Yes.
During this reaction from A to B, during this reaction from A to B, which gives C, it gives ATP. During the reaction, you understand my point?
Professor,
Yes.
isn't this reaction anabolism? So it consumes energy.
Where is it?
A plus B.
Remember, we discussed this last
Metabolism.
time that we see a reaction in totality. Remember, we discussed glutamine example.
Yes, I remember.
Yeah. So in totality. Okay, I can give you a simple example of this. You know, honeymoon child. You know this example, this word, this terminology?
No.
Because you all are unmarried. Huh? So, okay. When someone got married, A married with B. They went to honeymoon to Paris. Okay. And then they come back and the wife is pregnant now. Now this pregnancy, this child, this pregnancy occurred during this phase, honeymoon phase. Is this clear?
Yes.
Sometimes it is called that, oh, this is our honeymoon child. You understand my point? This is a new term generated, just to make point clear to you. So when energy comes during the reaction, this is called substrate level phosphorylation. Substrate level phosphorylation. For example, 1,3 bis phosphor, bis mana 2, di mana 2, phosphate mana phosphate, glycerate. You remember? 3 carbon compound glycerate, 1, 2, 3, glyceraldehyde. This is carbohydrate. This is OH. This is OH and this is CH2OH. 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 Clear? I can write this like this. Huh? This is H here and H here. CH2OH. Clear, guys? If you add phosphate here, 1, 2, and 3, this is phosphate here, and here phosphate here. So this will be 1,3 bisphosphoglycerate. 1,3 bisphosphoglycerate. Is this clear?
Yes,
Yes,
clear.
clear.
This enzyme, phosphoglycerate kinase, will act on it, remove phosphate from position number 1, add this phosphate to ADP, add this phosphate to ADP, and then this 1,3-bisphosphoglycerate is broken down into 3-phosphoglycerate, and ADP is converted into,
ATP.
we got energy, we lost energy.
Create
Got it.
energy, release.
We got energy. Huh?
Yeah.
During the reaction, final product is that we received energy. ATP is formed. But this ATP is formed during the reaction. This
Not
ATP is
result.
formed during
Yes.
the reaction.
The main
Clear?
reaction for biophosphogyrestrate.
Yes. So this is called substrate. This is substrate. Huh? This is substrate. Level phosphorylation. Clear? Another example.
Yes, okay.
Another example. Phosphoenol pyruvate. Phosphoenol pyruvate. Pyruvate is a 3 compound. It has 1 phosphate group. Pyruvate is a 3 carbon compound. It has enol group, 1 phosphate group. This phosphoenol pyruvate will be acted by enzyme pyruvate kinase, will remove its phosphate, and will add this phosphate to ADP. This ADP will be converted into ATP, and the remaining, what will happen remaining? Enol pyruvate will be remaining. Enol pyruvate will be remaining. Enol pyruvate is here. Clear?
Yes, okay.
Yes, clear. And
Substrate level phosphorylation. Another example of this substrate level phosphorylation. Mana, during the reaction, when you are traveling, you are earning money. Clear? Third example number 3 is succinyl-CoA. Succinyl-CoA. Now here in the first two examples, they already had phosphate with them. The substrate already had phosphate with them. You got this point?
Yes.
The phosphate is already present in the substrate. But here in the third example, substrate is not present in the substrate, but the reaction is such that succinyl-CoA will adhere 1 phosphate, it will take this 1 phosphate from cytoplasm, add to ADP, and will convert into succinic acid, and ATP will be formed. I can give you the example of this. An enzyme is succinate thiokinase. Is this clear?
Can you repeat that, Doctor?
Yes.
Can you repeat the last step?
Okay, I will repeat it. These 3 are the example that there is an Eid day. Eid malum Eid.
Yes.
You give money to the children. You give money to the children or not?
Yes.
Or you keep all money with yourself? Okay. Now, on Eid day, these are the 2 examples. You give money to your younger brothers, younger nephews, children, and this is your own money. This is your own money. You have this money. You have this 5,000 real, and you distribute it among the children. This is the example of this one. Clear?
Yes, clear.
These 2 are the example of this one. You have your own money. Glycerate has its own phosphate. Pyruvate has its own phosphate. Okay.
Okay.
Example number 3 is very smart example. Example number 3, that here is you are here. This is Eid and this is children. But you are maskeen, mafi fulus. But you are very smart. You took money from your mother. You took money from her and gave it to the children. You don't have anything yourself. Mafi ay shay, mafi wise fulus. You understand my point?
Yes, so clear.
You
That's
took money
clear.
from someone, from your baba, from your elder brother. Wallahi, brother, give me. You know, this is the position of my state. The people, the children will say, "Oh, you are our elder doctor brother. You don't have loose." You understand, my friend? So
Yeah.
now this is the position. So please give me some money so my position will maintain. So you took money from others and give it to the children. Is this clear? Here,
Yes.
succinyl-CoA is you. Inorganic phosphate is the mother, is the flus which you took from somebody else, from brother, from mother, from father, from anyone. And ADP are the children's. ADP are the children's. Clear?
Yes.
Yes, clear.
Is this clear? So it will convert into ATP. Here the same situation is here. This is ADP are the children. But here you are rich. Here you are rich. You have your own phosphate. So you gave him your own phosphate. Here you are too much rich. You have your own 2-phosphate. Is this clear?
Yes.
Yes, clear.
Sometimes the substrate is rich, has its own phosphate. Sometimes the substrate is not rich, but very clever, very intelligent. He knows how to move. Clear? He enjoy the life on the expense of other money. You would have sometime friend like this. Understand my point? So he is very smart. He enjoys the life. He gave fulus to maskeen. You understand my point? But not his own fulus. He will take money from you and give it to somebody else. Is this clear?
Yes.
All these are the example of substrate level phosphorylation. All these are the example of substrate level phosphorylation. The first two are the example of glycolysis, and the third one is the example of TCA cycle. TCA cycle. Guys, is it clear or not? Because I'm going very slow, okay?
Yes, that's clear.
You know my style, I'm a little bit fast, but now I'm very slow because these reactions are very important. Concept is very important. If you understood this concept in the beginning of the year, rest of the chemistry will be easy for you. Human body reactions will be very easy.
Can I ask a question?
This is the core lecture. Yes.
The chemical reactions, is it anabolism or catabolism in this?
Again, the reaction is giving energy or taking energy. These reactions producing energy or using energy?
Giving energy.
Giving energy.
Energy, that is, energy is giving. So what is that? Delta G is positive or negative?
Catabolism.
It's negative.
Positive.
See? It will give you energy. Clear?
Yes.
Yeah.
Exergenic.
Okay. Okay, guys. Now see here. This is the example. Example number 1. So can anyone explain this example?
This is the same example you just now explained.
Same example. Yes.
Yeah, so you have phosphate on the carbon number 1 and number 3, biphosphoglycerate. Okay. One of the phosphates comes off with the ADP and becomes ATP, and you left off with only 3 biphosphoglycerate.
Exactly. So this phosphate.
Is there anything else?
Good. Excellent.
Okay, alhamdulillah.
Now one more thing. If you see there are 2 phosphate, one phosphate has this sign. Another phosphate has this sign. Clear? Whenever you found this sign, this phosphate like this, so then you need to understand that this is a weak bond and this phosphate is a labile phosphate. Whenever there will be chance to move phosphate, this will move first, okay?
Okay.
So in 1,3-bis phosphoglycerate, position number 1 is a labile one and it is easily be removed. So this phosphate will go out, add to the ADP and come to make ATP. Clear?
Yes, clear.
Yes.
Okay, so this is the example of 1,3-bis phosphoglycerate converted into 2-phosphoglycerate, 3-phosphoglycerate, and then ATP is formed. This is, and we have same other example as well, like pyruvate kinase and succinyl-CoA. Okay, then we have another type of phosphorylation. The another type of phosphorylation, we call it oxidative phosphorylation, or sometimes we call it phosphorylation in the electron transport chain, ETC. ETC is also called respiratory change. Respiratory change. Why we call it respiratory change? Because in respiratory system, what happened in the respiratory system?
Oxygen comes to the body.
The involvement of oxygen, excellent. So if you remember, there are two kinds of the respiration in the body. One is respiration at the lungs and environment level. You take oxygen from outside from environment, it goes to the lungs, and then from the lungs, carbon dioxide come out to the environment. Clear?
Yes.
And second is that this oxygen goes to the cell, and from the cell, it enters into the electron transport chain where energy is produced. This is called cellular respiration. Cellular respiration. So how many cells are there in the body?
A lot.
How much a lot?
2 million, I think.
2 million. 2 million is in the
37 trillion.
Yeah, I think trillions.
trillion.
37 trillion, I guess.
2 million and 37 trillion. Maybe more than this, but not 2 million. Huh? It said maybe 75 trillion or something like that. So it is in trillions. Million is in meow, meow, meow. Not even not in meow, meow. Meow, meow have too much cells. Maybe in the cockroach. Even in the cockroach, maybe have too many cells. So it's at 75 trillion cells. Now see in the cells there is no use of oxygen. That's called cellular respiration. Cellular respiration. Clear? So there are three names: oxidative phosphorylation, electron transport chain, or respiratory chain. Clear, guys? So what we saw today, we saw the substrate level phosphorylation. Now let's move to the oxidative.
I just have one question.
Oxidative phosphorylation. Can somebody read this slide?
Okay, I'll read. Oxidative phosphorylation, a process by which ATP formed as electrons transferred from NADH to FADH2, 2O2 by series of electron carriers, carried out by enzymes and electron carriers located within inner mitochondrial membrane, electron transport chain, respiratory chain. I have a question, Prof.
Yes.
ETC and RC is the same thing?
Yes.
Okay.
Yeah, they're different now. So what happened now? What happened now? You have carbohydrate. You took carbohydrate from Alsia, fat, and protein from Hashi. Then we burn it. When we burn it, so step number 1 was energy and substrate level phosphorylation and ATP. Is this clear?
Yes, clear.
This was one process that we discussed. This was one process. Step number 2, what happened that during this process of burning, one more thing is released. And that one more thing is hydrogen. Clear?
Yes, clear.
This hydrogen is released. Now hydrogen is very mushkila. You cannot leave him free. Why you cannot leave him free?
Is it radical?
Yes, but why you cannot leave him free? Yes, it's a radical, free radical. Yes, good, excellent.
Free radical
Number one.
will cause damage.
Huh? Yes, go ahead, please.
Because free radicals will cause damage to the body.
One is
So
the
you can't
free
leave it
radical because it's a free radical. Yes, very good.
Irreversible
And
damage?
what it will do further? It will change the
Hydrogen acceptor, I think.
it will change the
pH.
pH.
Oh,
When the pH is changed, and what is pH? pH is negative log of hydrogen ion concentration. You remember this? And for the body function, you need normal pH.
7.8.
Otherwise, as is mentioned, there will be free radical injury. Is this clear?
Yes.
So you cannot let this hydrogen be free. So then what will happen? If you cannot let hydrogen be free, and in the oxidation of carbohydrate, fat, and protein, millions of the hydrogen are formed, then what will happen? So then what you have? Okay, I'm removing all these things. Huh? So what you have? You have hydrogen
acceptors.
acceptors. Very good, very good, very good. You have hydrogen acceptors. You have hydrogen acceptors. And there are two kinds of hydrogen acceptors. One is
fat.
NAD and another is
Fat. FAD.
they take two hydrogen, one as attached, one as free, as a radical form. They take two hydrogens, so NAD and FAD. So when they take hydrogen, they become reduced. So for example, this is A and this is AH. Which one is oxidized and which one is reduced?
The left one
A
is
oxidative.
A oxidized.
A
And
is oxidized and AH is
Reduced.
reduced. Very good. So these hydrogen acceptors, they are basically coenzymes. And when they receive hydrogen, they become reduced coenzyme. Clear?
Yes, clear.
It's clear.
And then these reduced coenzyme, now the story starts. These reduced coenzyme will go to mitochondria. If they are already present in mitochondria, maafi mushkila. But if they are present outside the mitochondria, then you have to take it to the mitochondria. Is this clear or not?
Yes.
Yes, clear.
If you are living in front of the metro station, maafi mushkila, you will come out and sit down in the metro. But if you are living far away from the metro station, so how you will reach to metro station? Through the
Bloodstream.
Stations.
Metro station,
The bus.
through the feeder buses. They are called feeder buses or shuttle buses. Clear?
Yeah, shuttle.
Shuttle service. Now in the College of Medicine, you are there in the College of Medicine and the metro station is far away. So from College of Medicine to the metro station, you will go through the shuttle buses. Clear?
Yeah.
Now these that reduce oil and fat, if they are present in electron transport chain, no problem. But if they are not present in the electron transport chain, then they will enter into the electron transport chain through the shuttle service. Is this clear?
Yes, clear.
Once in the electron transport chain, what will happen? They will release this hydrogen. Once they release this hydrogen, NADH will convert into NAD and FADH will convert into
FAD.
FAD, or we say that now they become oxidized. Now they become oxidized, and hydrogen will enter into the electron transport chain, and it will move in the electron transport chain to release energy, and this energy will cause the addition of inorganic phosphate from the cytoplasm to ADP to make ATP. And what is your idea? How much energy it should be that it will add one inorganic phosphate to ADP? More than
7.3.
7.3 kilocalorie.
7.3 kilocalorie. Is this clear?
Yes, clear.
So this process from here to here is oxidation, and this is phosphorylation. That's why we say oxidation and phosphorylation, they are joint processes, side by side. This is oxidation on the top and this is phosphorylation down, and they are joint process. Mana, you take food, you burn food, the burning of the food, this is burning. Burning will release energy, and this energy will be, this should be quite enough that it will add phosphate to ADP to make ATP. So these two processes are joint process, sister and brother processes, oxidation and phosphorylation. Is this clear?
Doctor, can we say it's anabolism?
Don't confuse yourself in anabolism and catabolism. Yes, yes. And in terms of energy, but in terms of breakdown of the product, it is catabolism. Clear?
Yes, sir.
Is this clear, guys?
Yes, clear.
Yes.
Okay. So just summarize it. What we discussed today. What we discussed today. So here, let me give you an example here, and then we'll finish it. Okay. This is, see, this is succinate. A 4-carbon compound. 1, 2, 3, 4. Clear? Clear, guys?
Yes.
Succinate dehydrogenase enzyme is there. It took 2 hydrogen from succinate, add 2 hydrogen to NAD, and combine NADH. And by itself, it is converted into fumarate. Halas?
Okay.
This NADH take 2 hydrogen, reduce NADH, take 2 hydrogen, enter into electron transport chain. By itself, it release hydrogen, and then by itself, it is oxidized to NAD. Again, come back here. Okay. This hydrogen is processed in the electron transport chain, releases energy, and this energy utilizes addition of a phosphate to this one. So this process is called oxidation, and this process is called phosphorylation. Because they are occur in mitochondria and electron transport chain, that's why we call it oxidative phosphorylation or electron transport chain. Clear?
Clear.
So what we discussed up till now, we discussed that metabolism is a highly organized reaction. In metabolism, we get energy. Energy is of 2 types. One is at the direct energy at the level of the substrate. That is called substrate level phosphorylation, where the reaction is so much fast, so much strong, that it releases that much amount of the energy that one inorganic phosphate attached to phosphate group directly and can make the energy, like this reaction. In other type of the reactions, during the metabolism, in addition to that direct formation of the energy, we also release the metabolic processes release hydrogen. These hydrogen are very dangerous. These hydrogen are carried out to the mitochondria by the help of hydrogen acceptor. These hydrogen acceptors are the coenzymes. When these hydrogen acceptors accept the hydrogen, they become reduced. There are two kinds of the coenzyme. One is NAD, another is FAD. When they take hydrogen, they become reduced NAD, reduced fat, or reduce NADH and FADH2. These NADH2 and FADH2 are entered into the mitochondria. If they are already present in the mitochondria, okay, good. If they are not present in the mitochondria, they will be transferred to the mitochondria with the help of shuttle pathways. In the mitochondria, the hydrogen are released again. NAD and FADH will oxidize to NAD and FAD. And this hydrogen will enter into the electron transport chain. With the series of the reaction of the electron transport chain, they release energy. And once they release energy, any reaction that releases energy more than 7.3 kilocalorie will cause the addition of phosphate to ADP to make ATP. Make ATP. Clear?
Clear.
Any confusion? Any confusion? Any question? Yes? NAD, no need. I didn't say NAD goes back. No need to go back. It can be remain there. It can be utilized there. Coenzyme, we have too much coenzymes. Cytoplasm has too much NAD. It will be utilized there again. Clear? It can be utilized in the mitochondria or it will be broken down.
Doctor,
Yes.
after the coenzyme are reduced, then they go to the mitochondria electron transport chain. I didn't understand after that.
You didn't understand. What you didn't understand?
What will happen after they go to the electron transport chain?
Electron transport chain, the hydrogen will enter into electron transport chain.
Okay.
Hydrogen will, after entering into the mitochondria, hydrogen will enter into the electron transport chain. And then electron transport chain, there are several reactions. We will see it later on. And these several reactions will give energy.
Okay.
Okay. So for example, I will give you one clear example. For example, this NAD, when it brings hydrogen to the electron transport chain, so this will go to one reaction, second, third, fourth, fifth, second, like 10 reactions. And then this will give release energy. Not all the reaction gives you energy. That much energy which adds phosphate. Only 3 reactions. Only 3 reactions will give you this much amount of the energy. So NAD, when NADH enter, only 3 reactions give this much amount of the energy that can add phosphate. So there will be how many ATP? 3 ATP. And when FAD enter into the FAD bring hydrogen and it enter into the electron transport chain. So only 2 reactions gives this much amount of the energy that can add phosphate. Clear?
Yes.
Okay, guys. Now your homework is that you go home, you are already in the home, open the book, Lippincott or Harper, whatever the book you want, and read this oxidative phosphorylation, bioenergetic and oxidative phosphorylation. And we will see each other in next class, inshallah. Okay, guys.
Thank you, Doctor.
Because the concept is very important. Next time we will discuss shuttle pathways. Shuttle pathways. Shuttle pathways. Your job. This picture is of Harper. So if you open Harper or if you open Lippincott, you will find this pathway. Your job is to study the electron transport chain and oxidative phosphorylation or bioenergetics. Thank you very much and best of luck.